Friends, in this video I am going to talk about stay-green traits and its significance in crop improvement. This video contains introduction, strategy for development of genotype with stay-green traits, physiological basis, importance of stay-green traits, genetic manipulation of these traits, case studies, idiotype, relationship between stay-green traits and grain yield, and conclusion. Coming to stay-green trait, the extended foliar greenness during grain filling under post-anthesis drought is nothing but stay-green trait. Stay-green sometimes refers to the heritable delayed foliar senescence character in model and crop plant species. This is one such trait, and genotypes possessing this trait maintain more photosynthetically active leaves than genotypes not possessing this trait. Genotype with stay-green trait is called stay-green genotype, may be partially to completely dominant trait. Stay-green, a partially to complete dominant trait, is a major component of post-flowering drought resistance in sorghum. Where, when and why stay-green traits are required. Stay-green is required especially in a drought environmental condition to keep greenness of leaves alive for longer period of time, especially during the green filling stage, to maintain or increase high grain yield, and stay-green results when plant's normal process of senescence is disrupted. Strategy of development of genotypes with the stay-green traits. It is keeping alive for longer is the fundamental strategy for increasing the crop yield, marker value or market value. Physiological basis of stay-green traits. Coming to components of stay-green traits in physiological traits, the key component determining stay-green are total plant leaf area, that is TPLA, with positive correlation at maturity stage, duration of leaf senescence, that is positive correlated with GLAM, that is green leaf area at maturity, then rate of leaf senescence negatively correlated with green leaf area at maturity, and branching, that is indicator of stay-green trait. Classes of stay-green traits. There are 5 ways of stay-green on the basis of time and duration of occurrence of senescence. First one is type A stay-green, that is delayed initiation of yellowing. All these 5 stages will be diagrammatically explained in the next slide. Type A stay-green means delayed initiation of yellowing. Type B is that initiates senescence on schedule but comparatively slow. Type C stay-green means it arises due to specific defects in chlorophyll degradation pathway. Type D stay-green is cells are dead. Type E means chlorophyll content remain same but enzyme activity is reduced. Here it is given in the central, we can find the normal one. In type A, it is delayed initiation of the normal rate. Then type B is normal initiation but slower rate. Type C, it is normal initiation rate but lessen in chlorophyll degradation. Then type D is rapid tissue death, and type E is enhanced greenness initiation and proportional rate as normal. The importance of chlorophyll in stay-green trait. Chlorophyll pigment is responsible for greenness in the leaves. This pigment involved in photosynthesis. The result of photosynthesis is biomass production. When where will be degradation during the reproductive stage, ultimate biomass production will be less. Chlorophyllase is responsible for degradation of chlorophyll to a chlorophyllide. When a mutants disrupt the activity of chlorophyllase, it is leading to stay-green phenotype. Is the biochemical pathway of chlorophyll degradation. When chlorophylls act on chlorophyllide, that will start converting to chlorophyll breakdown. But whenever it is not broken down like this, means the chlorophyll will remain active for a long time. Relation of nitrogen with the stay-green traits. The first and foremost factor was the physiological or the biochemical mechanism. The second was the chlorophyllase activity, and the third is relation of nitrogen with stay-green traits. This is chemical nature of chemical nature of degradation. In this, chlorophyll pigment is consist of nitrogen, magnesium and other constituents. Stay-green can be viewed as a consequence of balance between nitrogen demand by the grain and nitrogen supply during grain filling stage. While nitrogen is the constituent of most of the proteins because it is involved in rubisco, and rubisco is involved in photosynthesis. Coming to importance of stay-green traits. Delayed senescence is useful only when it contributes to increased yield. Nitrogen uptake during grain filling is higher in stay-green genotypes than in senescent hybrids. In some crops it leads to drought resistant as well as it leads to lodging resistant. In horticulture crop, it increases horticulture or market value. Several approaches have been utilized to manipulate hormone level or responses in transgenic plants to reduce the incidence of post-harvest yellowing. It extends the shelf life and helps in long-term transportation. These are the genes and traits responsible for stay-green traits. For stay-green, SGR, NYE1, SID. For that mutant gene is stay-green, that is SGR, and the function is binding LHC1, that is leaf chlorophyll content 2, and catabolite enzyme stabilizing catabolic complex. Then chlorophyll B reductase, that is responsible from the gene NYC in rice and Arabidopsis. This function in ferredoxin or NADPH dependent two-step conversion of chlorophyll B to chlorophyll A. Then null gene, this occur in rice but not in Arabidopsis. Then HCAR, that is responsible for cell death, not stay-green. MG dechelatase, this is functional in removal of Mg from the macrocycle. Then pheophytase, pheophorbyde A oxygenase. Then RCC reductase. These are the enzymes and certain responsible genes which are functional in converting or making or breaking down the stay-green traits. SGR, NYC1, PPH and ACD2 are coordinately regulated at the transcriptional level in Arabidopsis. RCC reductase assemble into a complex with SGR LHC2 prevents photooxidative damage. Coming to methods to be stay-green. Regulation of chlorophyll metabolism, photochlorophyllide oxidoreductase enzyme helps plants to yield chlorophyll independent of life. Carbon capture, nitrogen remobilization and stay-green C and N export cases ceases in the terminal phase of leaf death, and hormones, transcription factor and stay-green. Reactive oxygen species and Virki NAC genes are responsible for the breakdown of stay-green traits. This is a pathway whenever the net import of carbon assimilation or carbon capture is more during the initiation phase, means that will give better carbon utilization in the later phases. In the same way, nitrogen remobilization is also enhanced when there is sufficient nitrogen uptake or import in the initiation phases that lead to functional stay-greens and a delay in the senescence. Coming to genetic manipulation of stay-green traits. First one is identification of QTLs for stay-green. Requirement for QTL mapping are suitable mapping population, reliable genotyping, phenotyping, linkage map construction, then statistical tools for that, and confirmation of the QTLs responsible for stay-green traits. This is just a procedure of mapping population development, which is a prime requirement in linkage mapping. The next step, that is overview of genotyping through PCR approach and gel electrophoresis. These are some of the stay-green QTLs identified for stay-green traits. First one in English blue glass, that is Festuca pratensis. In this identified QTL is recessive nuclear allele SID. Then in sorghum STG1, STG2, STG3 and STG4. In Arabidopsis recessive gene FIW mapped on chromosome 4. In rice recessive mutant SGRT on chromosome 9. In rice only 46 male QTLs for 6 traits are localized in 25 chromosomal regions, and in wheat QSG.BHU1A, QSG.BHU3B and QSG.BHU7D. Coming to introgression for stay-green QTLs through molecular approach. This is marker-assisted selection, through which marker-assisted backcrossing helps in continuous incorporation of the QTLs into the target population. This is a mechanism how foreground selection, recombinant selection and background selection is done to identify the genes and mapping the genes in a given population. Coming to different case studies, introgression of stay-green trait into Kenyan farmer preferred sorghum variety. In this, a study was conducted by ICRISAT scientists, where the parental line E36-1 has 3 stay-green QTLs, SBI-1, SBI-7 and SBI-10 located at various chromosomes. Here E36-1 is donor parent and OCHUT is a recurrent parent line. This is the overall procedure how they have done the transfer of successful QTLs into the plant. Coming to idiotype of genotype with stay-green trait. For idiotype for stay-green trait, plant should have spread and deep root system. Genotype with stay-green trait should slow the rate of leaf senescence, then delay the onset of leaf senescence, and genotype should have more total plant leaf area. The relation between stay-green trait and grain yield. Grain yield was correlated positively with green leaf area at maturity and negatively with rate of leaf senescence. That is, GLAM is directly related to grain yield, and rate of leaf senescence is inversely proportional to 1 by grain yield. Concluding stay-green traits, several stay-green QTLs have been identified in several crops like sorghum, wheat, rice, etc. By hybridization program, it is enabled to transfer identified stay-green QTL to the genotype having good economic background. Incorporation of stay-green traits in genotype will increase the ultimate grain yield and market value, which will contribute to our national economy. The stay-green traits constantly promote the good harvest as well as further requirement of the farmers. So it is one of the idiotype approach in any crop. Thank you. Hope you like this video. Don't forget to subscribe my channel. Thanks once again.